Hello, good afternoon. My name is Doug Huang, and today I'm going to be presenting on the research paper titled E-cigarette vapor enhances pneumococcal adherence to airway epithelial cells. A brief overview of the paper. The goal of the following experiments was to discover how e-cigarette usage increases risk of pneumonia. The methods for these experiments included live human volunteer testing between non-vapors and vapors, and in vitro cells purchased from a lab, tested with e-cigarette vapor extract, antioxidants, and PAFR blockers. The results show that PAFR expression was significantly increased in cells exposed to ECV and had a correlation to bacterial adhesion. Future experiments can explore the methods of oxidative stress, and hopefully, for those who are reliant on e-cigarette vapor and smoking, find a healthy alternative to vaping. An introduction into the paper and the first experiment. Platelet activating factor receptors, PAFR, are membrane receptors that are activated by the platelet activating factor. Many epithelial cells and leukocytes have platelet activating factors to enhance the immune response towards injury or disease. Damaged or infected tissue causes the release of PAF. Platelet activating factor activates platelet activating factor receptors, which causes the chemotaxis of platelets and other leukocytes. Activation also causes the release of more cytokines that activate an even stronger immune response. This is a representation of a PAFR. So the ligand, in this case the platelet activating factor, would bind with the PAFR and induce an immune response within the cell, causing the production and release of cytokines that causes a cascade of immune responses. For the sake of pneumococcal adhesion, the bacteria adheres to the receptor itself, and when the receptor is inducted into the cell, the bacteria can infect the human cell. For the volunteer testing methods, human volunteers were split up between an experimental group and a control group. The control group consisted of adults who had no exposure to vaping or smoking, and the experimental group consisted of adults who vaped at least once a week and had not been exposed to tobacco cigarette in 3 months. Test subjects were asked to vape normally, and samples were taken an hour later through a curette, which scraped the nasal epithelial cells from tissue. These cells were washed in PBS, otherwise known as phosphate buffered saline, which is used to maintain a steady pH and have ion concentrations similar to the human body. These cells, the control group and the experimental group, were tested for PAFR by using PAFR targeting fluorescent antibodies. This is an example of a fluorescently labeled antibody. What it is designed to do is to adhere to its target and give off a special wavelength when a laser is shined on it. The next slide will review the next section of this experiment. It used secondary fluorescent antibodies that attach to the primary fluorescent antibodies to enhance the initial signal. These cells were then tested with flow cytometry. In flow cytometry, sample fluid is injected into an instrument. A laser shines through these samples and the fluorescent marked samples emit different wavelengths of light. A computer can examine these emissions and count the numbers of fluorescent examples. In this experiment, there are 3 levels of fluorescence. 1 to highlight the PAFR, 2 to highlight total number of epithelial cells, and 3 to highlight the PAFR even more so. The cells were counted using E-cadherin. E-cadherin is a fluorescent type of the cadherin protein. Cadherin is a transmembrane protein that can bind to cells. This type of cadherin was used to identify the total number of epithelial cells. The experimental group later used software to analyze the amount of PAFR expression and adjusted for isotypic control and nonspecific immunostaining by using rabbit IgG. These are the results of the live human testing. So if we take a look at figure A, the y-axis is about PAFR expression, and the x-axis reviews the control baseline and the vapors before taking a hit. As you can see from the left side, there is no statistical difference. The p-value is 0.18 between the control and the vapors baseline. In terms of statistical value, an accepted value for the p-value is less than 0.05, which means that there is a 5% chance that this occurs by chance. So there is an 18% chance, which means that there is no statistical difference between these two groups. In figure B, there is a significant difference in PAFR expression and the vapor's epithelial cells before and after vaping, which implies that PAFR expression is greatly increased due to the pressure of the oxidative stress caused by the chemicals in the e-cigarette vapor extract. This next slide is an introduction into the second significant portion of the experimental processes in this paper. The ultimate goal for this experiment was to know if pneumococcal adhesion and PAFR expression are related, and if the mechanism for pneumococcal adhesion is caused by e-cigarette vapor extract. On the right-hand side, you can see that the in vitro cells were split between three different types: alveolar cells, bronchial epithelial cells, and nasal epithelial cells. There were controls for each cell type, and the alveolar cells were exposed to nicotine and e-vapor cigarette extract. The bronchial epithelial cells were exposed to antioxidants, and the nasal epithelial cells were exposed to PAFR blockers. All this contributed to the greater picture to show that vaping causes oxidative stress, which causes the expression of PAFR receptors, and these PAFR receptors increases the chance of being adhered to by a pneumococci. The next slide is going to detail the in vitro test methods. To assess bacterial adhesion, cells were incubated with pneumococci and the CFU per milliliter was assessed. CFU is short for colony forming units. To assess the role of oxidative stress, cells were incubated with NAC, an antioxidant, then were exposed to pneumococci. To assess the role of PAFR, a PAFR receptor blocker was added to an adhesion assay. These are the results from the in vitro tests. A single star represents a p-value of less than 0.05, which is statistically significant already. A double star represents a p-value of less than 0.01, and a triple star represents a p-value of less than 0.001, which means that any of the tests indicated with a single star and up are extremely significant to the results that we are trying to understand. For figure A, we're looking at alveolar epithelial cells that were exposed to nicotine at 3 different concentrations and e-cigarette vapor extract. From this table, the first thing that we know is that a higher percentage of nicotine causes an increased amount of bacterial adhesion. Secondly, e-cigarette vapor extract does have a significant role in increasing the amount of bacterial adherence. For figure D, the PAFR blocker CV3988 was employed. For this example, one group was exposed to 5% nicotine and e-cigarette vapor extract, which in figure A shows the highest increase in pneumococcal adhesion. And from this figure, you can see that having a PAFR blocker significantly decreases the amount of pneumococcal adherence to alveolar epithelial cells. The next slide reviews the results for the bronchial epithelial cells. Once again, the experiment is comparing the colony-forming units count and seeing the increase in bacterial adhesion in epithelial cells. One group was exposed to 5% nicotine with e-cigarette vapor extract and without e-cigarette vapor extract. Both have an increase in bacterial adhesion. Another group was incubated with NAC, which is an antioxidant. This shows from the results that NAC significantly decreased bacterial adhesion. So what can be inferred by this test is that because e-cigarette vapor extract causes oxidative stress due to the toxic chemicals within the vapor, if an antioxidant is applied to this exposed group of cells, then the negative results of the e-cigarette vapor extract can be limited. But overall, this shows that oxidative stress is one of the key factors in causing pneumococcal adhesion to human epithelial cells. This last slide for the results details very similar findings from the previous experiments, except these were conducted on human primary nasal epithelial cells. On the y-axis, you have the amount of colony-forming units, and on the x-axis, you have two groups, one exposed to 5% nicotine and e-cigarette vapor extract and without e-cigarette vapor extract, and another group exposed to 5% nicotine, e-cigarette vapor extract, and the PAFR blockers. This shows that the PAFR receptor is being co-opted by bacteria because this PAFR blocker inhibits the bacteria from co-opting the PAFR receptor. Therefore, there is a statistical significant difference between bacterial adhesion when the PAFR receptor is not able to be co-opted. Lastly, discussion of the final results. Both 5% nicotine-free and nicotine-containing e-cigarette vapor extract contained metals that could induce oxidative stress. Oxidative stress is the mechanism that increases pneumococcal adhesion. We know this by using the antioxidant test with the human epithelial cells. Thirdly, it is also clear that PAFR is co-opted by pneumococci because the PAFR blocker prevents its adherence. And the final conclusion is that vaping leads to an increased risk of pneumonia because it has chemicals that elicits an immune response that increases the expression of something that can be co-opted by bacteria. And that is all for my presentation. Thank you.